Welcome everyone to this week's Data Science Seminar Series talk. I'm David Bader and I direct the Institute for Data Science at the New Jersey Institute of Technology. We're very pleased today to have our speaker, Aydin Buluc from the Lawrence Berkeley National Lab and the University of California, Berkeley, to speak on large-scale graph representation learning and computational biology through sparse matrices. Aydin Buluc is a staff scientist and principal investigator at the Lawrence Berkeley National Laboratory, LBNL, and an adjunct assistant professor of EECS at UC Berkeley. His research interests include parallel computing, combinatorial scientific computing, high-performance graph analysis and machine learning, sparse matrix computations, and computational biology. Previously, he was a Luis W. Alvarez postdoctoral fellow at LBNL and a visiting scientist at the Simons Institute for the Theory of Computing. Dr. Buluc is a recipient of the DOE Early Career Award in 2013 and the IEEE Technical Committee on Scalable Computing Award for Excellence for Early Career Researchers in 2015. He is also a founding associate editor of the ACM Transactions on Parallel Computing. Thank you, Aydin, for speaking with us today. And I'd like to let the audience know that if they have questions, please use the chat during the talk and we'll get to those questions.
Thank you very much, David, for this kind introduction. And thank you for generally inviting me to give the seminar. I should probably change the last sentence of my bio now that I'm relieved from my editor duties after 7 years. Welcome, everybody, and thank you for dialing in or zooming in is probably the verb these days. Today, I'm going to talk about, as David said, graph representation learning and computational biology through sparse matrices. And you might be wondering what are sparse matrices. One of the most famous people who have worked on sparse matrices is perhaps Harry Markowitz, who won the Nobel Prize in economics for portfolio theory. Not necessarily for his work, maybe in sparse matrices, but he has been a founder of the field. And in 1950s, he made this quote that says, "I observed that most of the coefficients in our matrices are zero, and if you carefully exploit that fact, you can do Gaussian elimination without spending time on the zero entries." That's essentially what he was saying. And for most people, a sparse matrix is something that's used in scientific computing when you have an input matrix and you're trying to solve AX equals B, and you can do a factorization. And there's decades of interesting research through graph theory and other combinatorial algorithms how to do this operation in the minimum amount of so-called fill and flops, which are the operations. And if you're solving for other systems through iterative methods or eigen systems, you're still going to be using mostly sparse matrices. And that's usually a fact because a fact that arises from the systems that are being modeled being sparse. But nowadays, what you might be able to find is the machine learning software you're using. For example, majority of PyTorch is running on sparse matrices whenever the models or the data is sparse. And we're going to be touching on special cases of those through graph learning. So the outline of my talk is, it looks a little long, but it's actually pretty coherent. I'm going to first describe an effort called GraphBLAS, which tries to standardize the operations in the language of matrices that we thought would be useful for graph computations, which are analysis of graphs. And then I'm going to go over some primary patterns of graph computations using linear algebra. Then we'll go into graph representation learning and computational biology with 2 examples each. So I already told you the standard use case of standard use case of sparse matrices in scientific computing. But the machinery, the technology, and the data structures, algorithms are applicable to many other fields, and that's the focus of my talk today. So the GraphBLAS effort, the talks for this has been happening for over a decade, and there's actually an excellent book by Gilbert and Kepner on graphs algorithms in the language of linear algebra. But the actual effort to come up with an API that would kickstart implementations is I attributed this to this 1.5-page document, which basically lays out the vision that we should have an open effort to define a standard. And you can read all about this in the GraphBLAS.org. And we usually run a workshop. I usually use the closest event that's on the calendar to the talk when I advertise this. And GraphBLAS at IPDPS is a month from now, so that's the right event to advertise at the bottom. So what does a GraphBLAS API look like? It looks really like at the bottom. You can think of it as this is a matrix-matrix multiplication, where you might be asking, okay, we've been multiplying matrices since we had calculators before computers even were well defined. Well, that's right. Except so here's the differences here. One is I don't actually define whether these matrices are sparse or dense or any particular shape. It is an opaque data structure, and the implementation is expected to exploit sparsity whenever it's present and store it and take advantage of it in its algorithms, depending on its internal parameters of what it makes sense. So that is actually my favorite definition of sparsity, which is not in the slide. That is, if the number of entries that are 0 are sufficiently high that it makes computational sense to take advantage of the sparsity, then it's a sparse matrix. So it doesn't notice that I'm not talking about something like needs to have a constant number of non-zeros on times the logarithm of the dimension or something like that. I'm just saying some economical argument that if you can take advantage of it and make it go faster than using it as a dense matrix, then it's a sparse matrix. So the other important things I want to highlight in this spec are there's a concept of a mask, which is this blue thing, which basically says, if you're doing this operation to multiply A and B, I'm telling you what entries of the output C I care and what entries of the output I don't care. So then the implementation can change algorithms or reorganize the computation to never do the work that are not needed for the entries that are so-called masked out. And then there's this extra additional interesting things. But the big deal is the semiring, maybe, which is that this is not restricted to the standard real field that we run our computations on whenever we define doubles or floats or B floats, 16s these days in machine learning. But you can effectively define any semiring, which is an algebraic structure like a ring, except that it doesn't have an additive inverse, and run the computation on that. And that comes really handy in running a lot of different computations with the same data access patterns, but at the end computing different things. Think of it like shortest paths primarily. So I've been asked many times to very briefly describe some of the patterns that happen in GraphBLAS. And here's the 3 fundamental patterns, I think. And the first one is sparse matrix times sparse vector. And in this example, what we're seeing is we're in the middle of an iteration. We have started our breadth first search on the 7-vertex graph from vertex 1. And now we have explored this 1-hop neighbors already. And our current frontier includes 2 and 4, which are green. And I'm going to try to explore the next frontier. And what do? I multiply my sparse adjacency matrix, in fact, the transpose of it. But it's really not important for the purposes of computation, with the sparse vector that only has entries that are vertices in the frontier. And if I do this multiplication in the right semiring, what I get is the output 3, 5, 7. And the entries there, the values there, really mark the parents, so-called. Where did I come from? Like I reach vertex 3 and my cursor says I came from vertex 4. Now you might wonder what are extra stuff that's going on. So this checkerboard pattern note here, which is vertex 1, is masked out. It technically is discovered if I multiply this sparse matrix with that sparse vector blindly. But since I have already previously discovered the first vertex, which is where I started from, and I have passed the previously discovered vertices as my mask, the runtime is free to never touch this entry. And how that happens is the subject of another talk. You can change the direction of the search by, instead of doing matrix vector product, you can say all the entries that are NASC masked out, I want to go through the rows of these matrix and see if I can reach the parents. And that's essentially automatically discovering the celebrated direction optimization in graph searches, also known as push-pull. So what I actually am suggesting is, with the right interface, some of the biggest algorithmic advances that have happened in graph processing, like direction optimization, would have been automatically discovered. So that's number one. And this is not just for breadth first search, of course, because a graph traversal is fundamental to many, I would say the majority of graph algorithms. Connected components, matching, ordering, we have used the same primitive to run these algorithms in large parallel computers efficiently. The second one is a simple generalization on surface, which is instead of doing a search from one vertex or following one frontier, I can follow many frontiers all at once. And that is becomes a sparse matrix times sparse matrix. Often the right-hand side matrix is not square, but it can be, depending on how much memory you have and how much parallelism you want to extract those trade-offs. And this is almost identical to the previous slide, except that I'm doing this from multiple source vertices. The applications of this can be anything where you do graph searches in parallel, but also some of those algorithms that do not seem like traversal-based algorithms at first glance, like triangle counting, actually fall into this primitive. So why is that the case? Here's how do you do triangle counting? One way of doing triangle counting with matrices, which is I split my matrix into two. In this example, I split it into L and U, but in practice, you can actually do it L and L transpose, in fact, and use a mask of the original matrix. So what I do when I multiply L and U is I am discovering all so-called wedges. Any path of the form 6, 5, 4, let's say, is a wedge. It's an unclosed triangle. And by finally seeing if there is the closing edge present 6 to 4, which is here described as an element-wise operation, but in my primitive GRB MXM, it's a single operation because I use the original matrix as my mask. Then I can just simply count triangles with one operation. And this is a traversal too. Why? Because I am actually discovering two-hop paths, which are the wedges. So that's actually a traversal. It's just a very limited depth traversal. Markov clustering, which is a celebrated algorithm for finding protein families from protein similarity networks, or actually used also in protein-protein interaction networks, is following the same pattern. There you are essentially a flow-based algorithm, depending on how you look at it, or a random walk-based algorithm. You're pushing probabilities to your neighbors, and everybody is doing this at the same time. And to accelerate and guarantee convergence, there is a process of inflating entries and pruning the network. And after, I would say, 10 iterations or so, you'll generally arrive into a graph that looks at all the way to the end, and you can just look at its connected components and declare them as your protein families. It's a general network clustering algorithm that works really well for protein interaction and protein similarity networks. And that's also boils down to the same pattern. And the last pattern I want to emphasize is sparse matrix times tall skinny dense matrix. This is a little new, even to me, and has been, I mean, we already knew about this thing. However, the only application I was able to think about it when I worked on it initially 7 or 8 years ago was eigensolvers. There's a family of eigensolvers, which are block eigensolvers. The most famous one is LOPCG, and they explored the eigenspace in blocks as opposed to just one vector at a time. But recently, this operation is becoming fundamental in graph neural networks and graph embedding. So there's the most canonical example of graph neural networks is perhaps the graph convolutional network, where each node aggregates their neighbor's feature vectors and combines it with its own feature vector and decides on a new feature vector. And this is trained with through backpropagation usually, which means there's a goal. The errors are propagated back and new updates that happen. You can do it without a goal, which would be graph embedding. You can blindly just aggregate your feature vectors of your neighbors at each iteration, like various algorithms such as node2vec does. But in both cases, the fundamental operation is the multiplication of a sparse matrix, which is the graph structure, times a vector of current embeddings at each node. All right, so this is the set of patterns that I really find interesting. Of course, you need a lot of the other parts in GraphBLAS, but these operations are usually the ones where the majority of the time is spent in a computation. Now I'm going to move on to graph representation learning. Especially graph neural networks is where I'm going to start, but then I'm going to talk about a different method called the graph kernel. So graph neural networks is many applications in sciences. And of course, if you're in a company like Amazon or Facebook, it has applications in recommender systems. In fact, beyond applications, it's somewhat fundamental to some of these companies' jobs, Pinterest's, Amazon's, Facebook's. But at the DOE lab, I'm not particularly interested in those. Surprisingly, it's very versatile. You can use it for proteins, particle physics, transportation, power grid materials, and many other little boxes that I didn't draw at the end. And one of the reasons this is so powerful is you can think of graph neural networks as a generalization of many successful networks. You can think of them even convolutional neural networks is a special case of graph neural networks where the connectivity pattern is just regular and nearest neighbors. So instead of only looking like a nearest neighbor connections in the convolutions, standard convolutions in images that we do, here we use convolutions in our neighbors. So we'll look at our neighbors, which can be connected arbitrarily to us, and define a convolution operation just like we would do in an image, except on an irregular structure. So what can we do this thing? Let's say we define this thing and we computed it. I want to first give an example of why it would be useful. If I map my graph structure into a latent space, then I can do one of the three things. I can classify my nodes, say that, okay, I have some nodes that I know what they're doing. I have labels for them, and I have some nodes that I don't have labels for them. So the classical use case of semi-supervised learning, and I can infer the unlabeled nodes to their latent embeddings. Then I can do graph classification, which says, okay, I want to say something about the whole graph, and that comes up often in chemistry and materials, where you have a family of many molecule structures, and you know certain atomistic properties of a small set of them, usually because it's expensive to run computational chemistry simulations to get those information for every molecule that is known to you. And there you can infer what you don't know through the graph neural networks. Or you can do link prediction about, okay, is this edge actually present or not? And that seems to be actually more flexible than you might think about. You might think that this is only useful for Netflix-like recommender systems, but it's actually very useful in a lot of scientific domains. For example, when we try to see if this particular track in high energy physics that we see from the experiment is a real track or potentially a noise in my experiment. Or we can say, is this link between two proteins that I see in the data, maybe this is by coincidence, or is this a real homology? And you can use this link prediction in a lot of these cases. And this slide, this figure is due to Peter Belikovich. So how do I train a graph neural network? The tricky part about training a graph neural network compared to training a convolutional neural network, which runs on independent images, is that graphs are connected structures. And that poses a lot of challenges. When I'm fed 100,000 images, I can treat each one of them as independent samples and do what's known as sample parallelism. More commonly, people will say data parallelism, but that's a more general term. So each give 1,000 samples to one processor, the next 1,000 samples to the next processor. And that seems to work very well because it has some inherent benefits of reducing communication. And that's a subject of a different talk, of course. Here, though, you cannot give different vertices of a graph to different processors and hope that these things run independently, right? So because they're connected. And also, what is your sample size? If I have 100,000 images, I can say, I'm not only going to run 1,000 of them. My batch size will be 1,000. And next iteration, I'm going to run another 1,000. And that seems like very trivial to do. Here, if I take a small subset of vertices as my initial batch, what happens after I train this neural network of depth, say 5, is from those initial set of vertices, I explore the aggregated 5-hop neighborhood. And I ended up touching almost all of my graph in many graphs that I can think of. So even though I think that I'm only working on 1% of my input data, I end up touching my whole data during the training process due to the connectivity structure. And that's usually known as neighborhood explosion, which makes mini batch stochastic gradient descent or many mini batch based other algorithms really hard to train on graph neural networks. And that requires sophisticated sampling algorithms. So you don't end up touching the whole graph whenever you do mini batching. For those reasons, today I'm actually going to talk about an alternative, which is a full graph training. Given a graph that have some initial labels, I'm trying to find next final labels based on the training objective. And I'm going to treat this whole graph without sampling. So my mini batch is n, the number of vertices in the graph. This has some benefits because it's actually faster training per epoch. It has some downsides, but because it's slower convergence per epoch due to known issues of full gradient descent versus stochastic gradient descent. But it's been shown that these things kind of cancel each other in the long term, and you get same time to reach the same accuracy. Mostly the same things that I mentioned before, written in text. For those of you who want to read, I probably got ahead of myself. And the algorithms I'm going to describe can be found in this link. They're implemented in PyTorch Geometric, which is one of the 2 leading frameworks for doing graph neural network training. The other one being DGL from Amazon. Both of them actually under the covers use sparse matrices, unsurprisingly. But let's get back to the depths of this operation. So what was it? I already gave you a glimpse of what it is doing, which is sparse matrix times multiple vectors, or tall skinny dense matrix, different name for the same problem. And this is the primitive used in graph convolutional networks. There are some graph neural networks that require different operations in addition to what we know as SPMF, which is sparse matrix times tall skinny dense matrix. And those graph neural networks are graph attention networks, for example, but I'm not going to talk about them today. Subject of another talk. So how does this process play out? You're given an input feature length, let's say 3 in this example. Of course, this is not realistic. It's usually on the hundreds. And at each layer of the GNN, you transform this feature vector into a different feature vector. It can be the same length. It often is not, to create certain information bottlenecks. And in the end, after L layers, you get your final embeddings that you can use to test to test your accuracy and propagate it backwards. And that's really analogous to what's happening in convolutional neural networks, right? There is a concept of a channel dimension, and after each convolution, the channel dimension actually changes usually. And here is the same case. What is the storage cost of this? So that is where the trouble comes from. You have n vertices and L layers. So I have to keep the embeddings of each layer so that I can backpropagate later, update them in backpropagation. So and assuming that each embedding is order f, when f is, let's say, 500, 256, these are reasonable numbers. It's probably going to go up as the graphs get larger proportionally. However, let's pick up what the people are running on these days, which probably is not the limit of this problem. 100 million vertex graph, 4 layers, not a particularly deep network, embedding dimension on average of 256. We're just gonna say, take the word average here, even though they can be different. We're looking at close to a terabyte of memory just to store the embeddings. Well, that doesn't even count for storing the graph itself, the adjacency matrix of the graph. So, and if the matrix is dense, let's say, not that dense, when I say dense, I'm only talking about 100 non-zeros per row. Well, there you have another 100 million times 100 edges, and that's probably going to be as big as what you're looking at here. So those are the reasons that we decided to do this in distributed memory. So here's the full process of GNN training, not that important. You have a forward propagation that does sparse matrix times tall skinny dense matrix times a really small dense matrix. This one is, I'm going to mostly ignore this because this is f by f. And as you know, f is in the hundreds. So we do, of course, analyze it, but it rarely changes the complexity because f is so much smaller than anything else in this. In the backward propagation, similar computation, except instead of multiplying with A transpose, I'm multiplying with A. And instead of multiplying with the current embeddings, I'm multiplying with its derivatives. I just called G for the derivatives of all the operations. And you can, of course, read the paper for its the derivations. And there's additional multiplication of the same kind, surprisingly. So what you're seeing here is the main workhorse is SPMF, as I've been saying several times so far. And the algorithms for that are, there's a lot of algorithms we can think of. And for the purposes of this talk, I'm not going to go over every single one of them, but any distributed matrix multiplication algorithm you can think of is potentially applicable with caveats. So what are they? One-dimensional algorithms that split the computational cube of n-cube space there, which is not completely dense again, because my input is sparse. One of them is sparse. And the 2D algorithms that slice it as fibers and 3D algorithms that slice it as blocks. You can think of all of them, and we did in the paper. But one of the most successful algorithms turned out to be what we call a 1.5D algorithm. And here's why I call it a 1.5D algorithm, other than the technically it actually fits the definition, but it's intuitively the 2D algorithms split both input matrices and the output matrix, as you can see on the right-hand side, in a 2D fashion. Here it's 3x3 grid, process grid. So I have 9 processors. I'm dividing it, the inputs all to 3x3 to the processors. However, in a 1.5D algorithm, I say, well, I'm going to divide the dense matrices in 1D and the sparse matrices in 2D. And you can read the paper about why we call it this way. The benefits of this approach is the dense matrices are already tall and skinny. So the dimension, the column dimension of them are already very narrow compared to the row dimension. Further dividing it only hurts arithmetic intensity. And doing it this way with 1D has the benefit of keeping the arithmetic intensity constant as we increase the number of processors. What is the downside? Well, if you run this algorithm or read the algorithm in the paper, you'll see that temporarily the storage costs of this is higher than the aggregate storage cost of the algorithm is higher than the inputs. What does that mean? Well, I had the size of A, the size of B, and the size of C before I started, let's say. And somewhere temporarily, this becomes size of A plus size of B times square root of the number of processes, or C in general, if I'm working on a P over C by C grid. And C can be up to square root of P. So that's the not the downside, the trade-off. But if you're strong scaling, you already have this memory, so it makes sense to actually use that extra memory to reduce both communication and increase the arithmetic intensity. And here are the results on some standard data sets. Here, what we're doing is a large scaling study on, I call it large because it was the largest at the time on this very challenging problem. People have done work on up to 8 or 16 GPUs. Here we're looking at hundreds. In fact, the paper includes results up to 700-800 GPUs, because then we end up on the experiments in the paper. You can find the cases where we run on 120 nodes with each having 6 GPUs attached. This is Summit. For those of you who don't know, this is the machine in Oak Ridge. The machine is a little odd in the sense that it's not very well provisioned for the computational power of the nodes. So the utilizing the network bandwidth is a challenge. But those details, again, you can find in the paper. This idea seems to work very well. The short of it is that by increasing C, let's pick a location, which is the replication factor of the 1.5D algorithm, where C equals 1 degenerates into a 1D algorithm, and C equals square root of P would degenerate into a 2D algorithm. In some sense, I'm actually getting much better scaling, and without it, I would not be getting any performance scaling on this problem. I would just be mostly plateauing. So that's good. And that's about the end of my graph neural network training part. And you can read more on the paper that I'm citing at the bottom, that was published in SC a couple months ago. So do I see any chats? This is a proper place maybe to stop and ask recordings. If there is slower convergence, do you want to manage, or should I read this, David?
Sure, you can read it.
Okay, thank you. If there is a slower convergence per epoch, how can training be faster? Because per epoch, runtime is faster. So it's a multiplication, right? It's per epoch, the full batch training, it's faster, but it takes more epochs. So in the end, it washes out. And the actual people who first reported this reality was a paper called ROC, R-O-C, from Alex Icon's group. And that's, I can't remember the full name, but if you do R-O-C, Alex Icon, you can probably find the paper where they show that it's mostly a wash. In fact, full graph training can be faster. Have you seen attempts to implement IPUs for GNN training as opposed to GPUs? IPU means what? I'm sorry for acronym problems. You can unmute yourself and.
It's a different type of processor architecture that I heard about from, I think, Michael Bronstein's talk a couple weeks ago.
What does it stand for?
I think integrated processing unit.
Intelligence.
Intelligent. Is this the kind of work that graph cores and whatnots are calling themselves, or I'm just speculating at this point? And the short answer is I don't know. But I have seen people trying this in FPGAs or any other hard architectures. In fact, I've seen people doing specialized hardware for GNNs. So a lot of things will probably come out soon. This is a very hot topic. Okay. Could you provide a good reference for the Markov cluster algorithm in its random walk-based version? It is, I mean, I don't know if people sat down and said, this is a random walk-based algorithm. The original publication, I don't think it shies away from the word random walk. The a cluster algorithm. I forgot the name. Macro cluster algorithm. There's a couple of papers that describe it, and the author is not shy about calling it a random walk-based algorithm. You can find the details on that. There's one paper that's probably the most mathematical, if you're interested, and that's the one that's published in SciM Journal of Matrix Analysis and Applications. Okay, let's get to a different kind of methods that are used for graph learning. These are kernel methods. Well, let's start with what a kernel method is in general. In many cases, our problem domain is separable by some simple object like a plane or a line. However, it's not separable directly in the space it's given to us. Right, here's one example from the famous book by Russell and Norwick, where if we look at the input sets in its original dimensionality, I cannot separate them easily with a linear boundary. But if I map it into a third dimension with the appropriate transformation, I can do that. Right, so in some sense, a kernel is a generalization of this idea. I'm given any object. It doesn't even need to be already in two-dimensional space or any feature space. It can be the graph, it can be the tree, it can be the string. And I define an appropriate comparison function in some potentially infinite dimensional space that allows me to classify and separate the classes easily. Right, and then there's this whole family of methods about how to do this without actually instantiating it. It's called the kernel trick. How can you do this without instantiating the data in the high dimensional space? You can do it by implicitly doing this operation during computation as opposed to explicitly generating the matrix. But that's not the point. I'm gonna, or at least not the point of my talk. I'm gonna talk about how to do marginalized graph kernels in high performance. So what are these? These are often the state of the art for classifying graphs as opposed to classifying nodes or edges. They still seem to be the state of the art. And the downside of these operations are that you have to define the kernel. So you have to choose a lot of among possible kernels that are fit for your problem. And here I'm going to talk about a random walk-based kernel, which is going to be defined on the product space in theory. Like if I have a graph A and a graph B, now think about the Kronecker product of the adjacency matrices of this graph, also known as the product graph. And I have these vertex 1 from graph A, vertex 1 from graph B. They become one vertex in the product graph. So I have cardinality n1 times n2 in the product graph, and I'm doing a random walk in that product graph. And the more I often hit the same vertex in different graphs, the higher the probability that these graphs are similar. So that's basically the intuition. And the math tells you that you don't have to actually ever generate this product graph. You can simply define a linear system that is based on this Kronecker product and never materialize the Kronecker product and solve this system by matrix vector multiplications using a standard algorithm like precondition conjugate gradient. And that's essentially what the main focus of getting high performance out of these operations. Now, I want to tell a little bit more about the graph representation learning angle. So the learnability of graph neural networks is generally higher than graph kernels, because in a graph neural network, I don't really need to define a particular kernel. So the user is relieved from that duty. And here I have to actually decide whether it's a random walk-based kernel or whether I'm going to use a different distance metric in defining my kernel. So that's essentially the user intervention that needs to happen. But let's get to the interesting part of high performance computing. Here's what I'm looking at the right is the whole system to solve to get similarities between two graphs. and I have the adjacency matrices of the graphs and the labels. In fact, this works with labels, meaning if it can give higher scores when it hits the same vertex with the same label, which is usually the atom in the atomistic definition of the molecule, right? So that's why it's marginalized graph kernel. The name comes from there. And it's defined on this Kronecker product, except that I'm multiplying with a vector. So I'm defining this operation as a holistic single primitive. Do the Kronecker product, multiply with a dense vector. So I am streaming to the small graphs, and each time I'm getting a small block of the first graph's adjacency matrix and doing the Kronecker product of it with a small part of the other matrix, sliding the inner loop slides across the whole of second graph, and then the outer loops slides on the first graph. And this way, I never actually bloat my memory with the full Kronecker product and do the whole thing in a streaming fashion. Now you can, of course, say, how can I get this faster? And one of the nice things we were able to do is a very standard trick in high performance computing called graph partitioning. So a graph partitioning can be repurposed to do sparse matrix ordering. Most graph partitionings, the output of it, you can use it to reorder your matrix, and it often will give you clusters of non-zeros. So this is really nice because I have here two little graphs, two little adjacency matrices representing graph 1 and graph 2. The operation that I'm doing is this big thing that's running on the product space. So I can do partitioning on these small matrices, which is very cheap and easily amortized due to my expensive big computation. So I'm only doing a reordering of these small matrices using graph partitioning, and that doesn't cost me much. And I increase or decrease the number of empty tiles during my streaming algorithm, and that really helps with the performance. So that's one big thing I want to focus on. And there's, of course, other tricks about bitmaps and so on, and that I'm not going to go over too much. In the end, when we compare this to the only two other tools that we could find, so this is a big unfair comparison plot. I apologize to the developers of the other tools, but we needed to compare with something. This is comparing with Cython and Python implementations. Of course, you can always beat such implementations, but the amount we could beat this was staggering even to me. So we're only running this in a single GPU on the whole drug bank or PDB datasets, protein database. And the whole computation that is computing graph kernels between every single pair of molecules in these datasets is about 3 minutes. Compared to close to a month in PDB for some of these implementations. So it was very surprising to me, and this operation is actually embarrassingly parallel. That's why we call it a high throughput solver. So it wouldn't be hard to generalize this to distributed memory, though I don't see too much point unless the data sets get larger. Okay, so this is the part that I'm actually done with everything I want to say about graph representation learning. So I'm going to talk briefly, maybe about 10 minutes, on computational biology and two nice applications of sparse matrix operations in computational biology. So the first one is this operation called DNA read overlapping. If you're in this field, or at least following the trends, the long reads from two new companies, not new companies, but maybe new for investors, Oxford Nanopore and Pacific Biosciences, have been revolutionizing de novo assembly. Why? Because the dominant paradigm was the short reads from Illumina, which are 100 to 300 base pairs. Even though they're very accurate, their errors were biased, meaning that they would make the same error in the same location of the genome, which makes it really hard to recover from those errors. And while PacBio and Oxford Nanopore datasets, by contrast, are significantly longer, which where I'm talking about 15 to 25 kilobases, like thousands, tens of thousands of base pairs. and compared to hundreds. They used to have very high error rates, meaning 15 to 25, and that is already coming down with new technologies to around 5 to 1% range. So what that shift makes is the paradigm, algorithmic paradigm for assembling these genomes is shifting back from the so-called De Bruijn graph approach to the overlap layout consensus approach, which is first find all the overlaps between your reads, because you don't have that many reads. When your each DNA sequence is 10,000 base pairs, you don't end up getting billions of reads like you would get for a large genome in Illumina sequencing. So you can actually create this overlap graph in memory. And once you identify them, you do some sort of layouting and get a consensus. That's the whole thing. So I'm going to focus on how I can find the overlaps between the reads. And it's going to start from this simple assumption that everybody knows, but it's good to know, good to remind the audience that don't work on this, that with the right mathematical model, you can find a length k of subsequence where you can say that if two reads were overlapping by some reasonable number of base pairs, and that's plotted in the x-axis, the probability of finding a matching k-mer between these two reads is plotted in each plot, right? So just think of one thing, right? So if my error rate was 15 here, and if I chose k to be 17, then I will catch, let's say, 90% of all the overlaps that are 2000 base pair length. And of course, it's going to go up. So with those analyses, you can choose an appropriate k. What does it tell you is that most of the methods are based on finding matches of either k-mers or a sub-selection of k-mers. Right? There's plethora of techniques about sub-selecting k-mers. They're called minimizers, they're called syncmers, and there's a large industry of them. But once you find k-mers, however method you use to create your subset of k-mers, and we have our own method, but that's not important because the method that I'm going to describe to compute overlaps is generalizable, or in fact, we already generalized it to any k-mer selection method. You can create this nice matrix called read by k-mers matrix. It's sparse because each k-mer only occurs in a certain subset of reads, if it's chosen appropriately, the k length. And there's additional information embedded here. And if I multiply this matrix with just transpose, which is computing the Gramian matrix AA transpose, then I get this output, which is read by reads matrix, again sparse, which says read 1 and read 3 has certain number of k-mers in common, and this is the number of k-mers that they're common if you write your semiring correctly, and this is the positions of those shared k-mers. Now you can run all sorts of sophisticated algorithms during this computation simply because the GraphBLAS API allows you to compute on arbitrary semirings and overload the addition and multiplication operations. And that's exactly what we do to keep track of where the seeds come from and how many of them and so on. And when we computed the distributed memory version of this thing at large scale, in fact, this is actually a bigger paper than just overlaps. It finds overlaps, plus it does some operation called transitive reduction, which is to transitively get rid of, to reduce the graph such that the transitive edges, like the redundant edges that go from A to C, when there's already a path from A to B to C, are removed. And that makes the final context generation simpler, so to speak. So we do both of them in this work, and it scales very beautifully, mostly because it relies on a lot of scalable sparse matrix primitives. Now you might ask the question, was this the only way, right? Can I not parallelize this operation in distributed memory by hand? And in fact, that is exactly what initially we had done. The first paper that I'm citing here is done this from a MPI bottom-up approach. How can we think of this problem from the scratch and think of mapping each k-mer to a distributed hash table and then trying to find its overlaps by some hash table matches, and so on. So that took years of a PhD student's effort. And compared it to the so-called dBALA 2D, where we directly called the sparse matrix multiply that was implemented in the combinatorial BLAS library, for example, which took weeks of effort of implementing, just figuring out software details of how to plug it in. And in the end, the weeks of effort was, at any scale on any graph was faster. And that's mostly because in the sparse matrix abstraction, we were able to find the right algorithms a long time ago, right? We know that 2D algorithms are better than 1D. For certain cases, 3D algorithms are better than 2D. And we know exactly in what conditions these algorithms become state of the art, in what concurrency and sparsity patterns. So we can just choose that one. It's been already implemented. Whereas it's really hard to think about different ways of parallelizing this thing in 2D, 3D, if you're looking at a direct Kramer and read abstraction. The final thing I'm going to talk about is actually very similar to the DNA read overlapping approach. And this one is protein similarity search, also known as many-to-many protein alignment. Just like in the DNA overlapping, where you have sequences from the same genome, hundreds of thousands of sequences that you're trying to find overlaps. Here, the problem is you have proteins, but these are not coming from the same genome in general. This is the whole set of proteins, for example, you can get from your large metagenome sample of tens of terabyte size, ocean sample. So you're looking at a gigantic input set, but the overlaps are usually very different than DNA. Why? Because first of all, there's no such thing as overhanging, right? So in the DNA, you can think of it as, I have welcome to Zoom's background, and I can't do things with my fingers without that getting it lost. But in general, if a protein is homologous, meaning it descended from the same ancestor, it is up to 70%, almost the length is usually 70% identical, right? There's no such thing as them only being 10% overlapping. So they're either going to be significantly overlapping or they're not homologous. However, the signal is much weaker here. And why is that? Because I'm not sequencing the same thing. I'm sequencing hemoglobin in a human from the analogous protein that carries oxygen to the leaves of a soybean. Okay, that's still called some globin. I forgot its name, but it's such a distant relative to hemoglobin that getting that homology is really hard. So you're looking at 20, 30 average nucleotide identity weak signal. To be able to catch that, instead of using exact k-mers, I'm gonna generalize my approach to create substitute for each k-mer using the historically accumulated knowledge that are encoded in these scoring matrices. That says, for all homologous proteins, L changed to G this many times, so the penalty of seeing this difference is blah. And this has been, these scoring matrices there are many of them, depending on your goal. Nice thing about this sparse matrix abstraction that I described in Bella is we can generalize this for the similarity search with the substitute k-mer concept. So I can introduce a new sparse matrix to the middle, S, that maps each k-mer to its closest 20 substitutes based on evolutionary distance. And instead of doing an A times A transpose like in Bella, I can do A times S times A transpose, which basically finds all k-mer matches between proteins, even if they're inexact matches, not exact matches, and still run the same distributed sparse matrix multiplication codes. I just need to define a new semiring, which takes about 2 lines of code. And in the end, even though we haven't used some of the most sophisticated techniques that state-of-the-art tools use, like reduce alphabets and so on, we are able to get competitive accuracy. So here's a recall precision line, and with different substitute k-mer numbers, These red squares are what Pastis gets, the protein similarity search tool that we developed. And you can see that this is quite Pareto optimal for what it is, right? Compared to other tools like MMSeq or LAST, maybe we have a couple more percentages to get here. But for an initial attempt, this is pretty good accuracy. And scaling wise, it's unprecedented. It's the thousands of nodes we're talking about, not cores. And by contrast, the only other distributed memory parallel tool, MMSeq2, stops scaling at around, I would say, 20 nodes and so on. So even though it's faster initially, the scaling curve is very flat after a while. So we end up being much faster at large concurrences. So in conclusion, sparse matrix techniques underlie computations from disparate fields like scientific computing, machine learning, graph analysis, and bioinformatics. And GraphBLAS already seemed to have the right abstraction with its flexible masks and semirings to be the default backend engine for these computations, as long as tools have been developed and maintained. And the large-scale data and extreme parallelism necessitates the need for distributed memory. And these trends seem to be staying here and will likely only get worse. And one of the tools we have at our disposal is communication avoiding algorithms. So we use those techniques on sparse matrices to mitigate some of the effects of these adverse technological trends. And I owe many of these slides to my co-authors in the works that I mentioned today. Of course, I have many more co-authors than this, but here's the list that I covered the work of. My research team is passion.lbl.gov. Even though the name sounds interesting, it comes from the passion flower, actually. And there's an acronym about it. You can check out all the publications and some of the videos, in fact. Thank you for your attention.
Thank you, Aydin, so much. I know that there's some questions in the chat, and I also have some questions. So we have a little bit of time where we could ask you those. Maybe I'll ask you a question, and then we'll go to the chat. And I would just through your talk, I was really pleased to see all of this computational biology work. I remember back almost 25 years ago, I did some early work in computational biology, assembling a mouse genome database. And at the time, working between parallel computing and the biology committee was somewhat of a challenge and really wasn't heard of. And now it seems that you're doing that ubiquitously. Could you talk a little bit about how you collaborate with biologists, and do they know GraphBLAS, or how do you do this collaboration successfully?
It depends on the person, biologist. Some biologists have computational backgrounds, and they of course value computational aspects accordingly. Some just want the tool to work and don't really care about the math and the computational pieces behind it. And the way I think, I'm not going to say that it's smooth all the time, but I think persistence is the #1 key, which is the least they want is for you to learn an interesting problem from them, go publish one irrelevant computer science paper, and never follow up and try to get that tool really working for the tasks they have at hand. And if you, I think they've been burned, the biologists have been burned by this approach in the past. So they try to avoid wasting their time educating computer scientists. So just to be, really caring about their problems and not taking shortcuts is the most successful approach.
That's great. So do you want to read the question in the chat from Alan Young?
Yes.
Or? Okay.
Yes, Alan, thank you. Very interesting talk. One of the slides shown a dense matrix that evolved to a sparsely separated key nodes. Oh boy. I don't remember. What might that be, frankly? I don't have a recollection of a dense matrix that evolved to a sparsely separated kernels.
Just to read the rest of the question for those on YouTube, is the notion an attempt to trace back to the source from the beginning nodes or close to it? Would this be applicable to find the key cells starting a cancer tumor, or maybe the start of a Big Bang? Thanks.
I think that's a bigger, more philosophical question than any of the pieces in my talk was trying to address. That's very interesting, but I didn't have an intention like that. Let's put it that way.
So maybe I could ask you another question. There's a lot of new types of processors on the market. We see highly multi-core Intel and AMD processors. NVIDIA this week announced in a few years this Grace ARM-based processor, and ARM is certainly of interest. There's RISC-V. What are your thoughts about architectural innovations needed to accelerate the sparse graph work that you're doing?
Yes, a wonderful question. I think NVIDIA is doing the right thing with the Grace approach. And if you watch Jensen's talk in the particular 3 minutes that he's introducing, he's showing the big bottleneck of feeding these GPUs. And that's exactly what we've been experiencing at a Machine Lab Summit, which has a lot of GPUs in a node and one PCIe, or in fact, one network interface card that that is the only highway exit to these. So they're trying to create this, as far as I can see, these processors will be able to feed the GPUs much more efficiently. If someone is going to do GPU computing in the future, that's clearly the path. In terms of general architectural trends, I try to avoid making guesses because the industry has its own limitations. I can talk just graphs, but industry has to support a lot of different computations on that space. So my feeling is that you can, for every adversarial architecture, there is enough human ingenuity that we can actually figure out how to make the best of those architectures with algorithmic advances. That's my personal take.
Well, Aydin, thank you again for giving this talk. It was just really great to see these sparse graphs being used in computational biology, and the work that you're doing at LBL, just really impressive. So thank you again.
Thank you, David, for inviting me. And you've been running this great set of seminar series.
Again, from the Institute for Data Science at the New Jersey Institute of Technology.
Double
Thank you.
Bye-bye.
Take care to everyone in the seminar.